Okay. You should see it. Okay. Small. Okay. Oh, that is okay. So looks good. I'm not sure if I can get away this zoom bars. I see all of that. Man, hey, every time something new, hey. Can't I just okay, whatever. It should be good enough. Okay, so welcome everybody. Today we will do mainly pharmacodynamics and trying to distribute some of the publications so that we everybody can start reading one of the publications and try to get some of the data out. So what I did to prepare the meeting, especially I did some literature research and evaluated some papers and made a list of priority papers. And then we have to select some of the publications and see what data is then relevant for the computational model. Okay, but first talking about pharmacodynamics, and then afterwards I will present a bit of the overview, what I figured out about lisinopril, and then we have to see what kind of data we want to use for the model. Okay. So what is pharmacodynamics? This is basically the second part of our of our computational model, which we want to build. On the one hand, we have the pharmacokinetics, which deals with the concentration time courses of lisinopril. And on the other hand, we're also interested in the effect of lisinopril, which is actually the pharmacodynamics. So what does the drug do to the body? And the main pharmacodynamic effect we are interested in is reduction in blood pressure. And okay, how this reduction in blood pressure happens with different concentrations of lisinopril. In reality, this is much more complicated, and okay, there's a lot of pharmacodynamic readouts which we could look at, but overall, I would say like the reduction in blood pressure is the simplest thing which we want to look at. And then if you combine this pharmacodynamic curves, which are the effect curves versus concentration, with the concentration over time curves of the pharmacokinetic part, you get this pharmacokinetic pharmacodynamic dose effect versus time. Okay, so how is this pharmacodynamic effect normally happening? So most of the times, the drugs interact with components in the body to produce a certain response. And most of these responses are due to proteins and binding of the drug to certain proteins and then changing the properties of these proteins. These are often receptors on cell membranes and the target tissues, or enzymes which are catalyzing certain reactions. And the drug is then basically binding to it and changing properties. It could induce activity, inhibit the enzyme, okay, change other properties. Like most common targets are G protein coupled receptors or channel linked receptors. Like for instance, with a G protein coupled receptor, you basically change a downstream cascade, often signaling cascade, via binding of the drug. With this channel linked receptors, you often change the transport properties of these channels. For instance, here you could imagine if the drug is binding, then less sodium is transported via the channel. Other targets are enzymes, enzymes or transporters. The enzymes, often the drugs are inhibitors or activators, and they change how much the enzyme can convert substrates to products. Here another example for transport protein, and then would just change this transport activity of sodium and potassium here. Okay, and this is basically what the drugs are doing, and there's a dose response in there, and often a saturation depending on the dose. Because you can imagine you have like only limited amount of these receptors, and at a certain point everything is all receptor is bound with the drug, and you have the maximum effect which you can expect in such a system. The effect is classified into can be classified into agonists and antagonists. The agonists basically increase the functional response, for instance, like in case of receptors, and then antagonists, they basically diminish the functional response. So agonists basically bind, and then you can like have like some, for instance, some activation. With antagonists, they bind, and then they basically block receptors and so on, and you can't have so much signal through the system or so much activity through the system. There's often like this one distinguishes between full agonists and partial agonists, and this says, okay, if the substance is capable or the drug is capable to show the full maximum effect, or if it's only showing a partial effect. In addition, you often find the words like inducers, which increase the synthesis or the protein amount of things. You see activators, which don't change the amount of enzymes or proteins, but they increase the activity, and inhibitors decrease the activities. And so, like, if you read the literature, and like often like you find this words, and like you should have a general understanding, okay, of what they mean. So how is pharmacodynamics characterized? Normally, this is done by a so-called dose response curves, and there you plot certain biological effect you're interested in, for instance, reduction in blood pressure, against the drug concentration. Often you find this in on semi-logarithmic or double logarithmic plots, and and these curves then result like are often sigmoidal or saturation curves. This dose response can be influenced by many factors, like disease, age, and so on. Some of them, but also presence of other drugs or other effectors can, of course, change this dose response curves. So normally what you're interested in is like that you get somehow from a time response, let's say, to a dose response. And I think that to understand this concept once, and it's pretty good, and then you understood like, okay, where the problems could be, and what things can arise from such curves. So often, like if you give a drug like eosinophil, you get this concentration time curve, and the concentration decreases over time. But then you see a certain response, and this response doesn't have to have the same behavior as the concentration. So you often have like a different curve shape in the response, and often also time delays, so that the response comes later than the actual drug you have given, and that you have like some nonlinear transformation. And this is often that the response looks different than the actual pharmacokinetics. Is due to this nonlinearity in the dose response curve. So you can imagine, like in the beginning, for instance, here you gave the drug, you have a pretty high concentration of here 10 milligrams per liter, and then you have to look in the dose response curve, okay, in which regime you are, and you're basically up here, and you're in the saturation part of this biological response. And so, like even if you change the concentration now, and you go down from 10 to 10 to 6 to 5. You don't change much in the dose response because you're in the saturation regime here. And so the concentration goes down, but you still show the maximum response because your concentration is basically above up here in the saturation range. So like look at the logarithmic axis, so like basically until 1, you don't the concentration of 1 milligram per liter, you don't see much response, and that's why this response curve is pretty flat here. And then you have like a range in the dose response curve. You see a pretty linear effect. And there you see also like a pretty parallel, let's say, parallel change in the response to the concentration. So concentration goes down, and the response goes basically very linearly down to the concentration changes. This is the mean range, and then in the end, you come in the range where again, even for very low concentrations, you still see a certain response, and this is here in the lower range. You still see like some remaining response. And I think it's just this understanding, okay, that you have like this nonlinear dose response curves, that these automatically result in basically in a transformation of this response time curves. If you have understood this and understood that there can be additional time responses, then often it makes sense why the biological responses have different behavior than the actual concentration responses, but it's basically just a combination of those response curves with the actual time dependency of their concentrations. There are two important concepts in this pharmacodynamic part. It's like called potency and efficacy, and basically these are the maximal value you have like of this dose response curve, and then the half saturation part. So the potency is the amount of the drug which is required to produce the biological effect, and this is basically the half maximal value, and the lower this half maximum value of the curve is, the higher the potency of the drug. So basically meaning like, okay, you need only like a little bit of drug and you have like a very strong biological effect, because the dose response curve like basically has half saturation at a very low concentration. Like for drugs with low potency, you basically have to give much more drug to get the same biological effect. But here, like it could be like two alternatives. ACE inhibitors, I don't know, lisinopril and ramipril, and they just have different potency, basically a bit different half maximal dose response curves or concentrations for the half maximum effect. And then you have different potency of the drugs to show the same biological effect. The second concept is efficacy. This is basically the maximum effect of the drug. And that means like, okay, how high this dose response curves are going. And this is a measure of the capacity to produce a biological effect. So basically the higher the efficacy, the larger the effect is. So those are the concepts. Okay, how much drug do you have to give? That's the potency. And what is the maximal effect which you can reach with a drug? This is the efficacy. And then the efficacy is one of the biggest problems in clinical trials, and why most drugs, or like it's a main reason why drugs fail in this phase 3 clinical trials, because the efficacy is just not high enough. So basically you see a certain effect, but the effect is not large enough, so that the drug, it doesn't really make sense to spend so much money on not having sufficient efficacy. Yeah, and here in this example, the drug with the highest potency has the lowest efficacy, and vice versa, but this depends on the drug and the actual system. What you often see is like desensitization to drugs, and this means like that you have a shift in the potency. It means you need like more and more drug to get the same biological effect, and effectively this means, okay, your dose response curves shifted over time. This is what I also meant on the slide before, that you have many factors which can change this dose response curves, like age, disease, and so on. And of course, if you shift this, you shift the potency and you shift the amount of drugs, and you shift also the change in the biological effect. And the most known, I think, is the tolerance. It's often like for drugs, you get basically like less effects of many of the drugs, and you just have to take more to get the same. Okay. So how can you describe this mathematically? Like what is often used is this simple dose response equations, which are very similar to this hill type equations, which we saw for the metabolism. As you basically have two parameters in this equation. The one is the Emax, which is the efficacy or the maximum response, and the second one is the C50, which is the potency, so the concentration to reach half maximum. We have also like this gamma in there, which is a steepness factor, and the higher the gamma is, the more sigmoidal on the curve is, and the sharper the shift is in this curve. Yeah, here are just a typical example, and you could then use this data and fit this parameters, the Emax and the C50, to this curve. Like for instance, here you can mention the maximal or the Emax is especially something in response unit. It would be here the percent of decrease in heart rate if you give this drug, probably a beta blocker or something like that. And it would be somewhere at around 35 or 40% would be the Emax. The half maximum would be then around 20, so it would be somewhere at 5 microgram per liter would be the C50 parameters to do a fitting. Okay, and then so often, you fit this very simple pharmacodynamic models to get the dose response curve fitted, and then you can combine that with the pharmacokinetic models, like for instance, build like a physiological based pharmacokinetic model, and then you have a simple dose response model for the pharmacodynamics, and you just couple these. The steepness factor, this gamma, is actually saying how fast is the switch here in the in this curves, and the higher the factor is, the faster the switching behavior is here. So you see for gamma of 5, you have like a very basically like a switch-like behavior, so that almost nothing happens below the half maximum value, and then you get like a very fast, almost full response if you have a little bit more drug. For other for the smaller gammas, you have like a more broader range of concentrations where you see a change in the response. Important concept in this context are beneficial and adverse effects. So maybe like similar things. You want to, if you give a drug and it has certain biological effects, and then some of these biological effects like are beneficial. It's often why you give the drug. You want to reduce symptoms or treat the disease and or block some pathway and so on, which is important in the disease. On the other hand, similar mechanisms can result in adverse effects. And you can imagine what you want somehow, that you have like this dose response curves for the beneficial effects and the adverse effects, like as far as possible apart from each other. It means basically if these curves are well separated, so like at some low concentration you have the beneficial effects of the drug, and at high concentrations you would have the adverse effect, then it's very easy to do correct dosing and avoid side effects. The closer these curves are together, you get basically an overlap of concentration ranges and so on, and so some people will have adverse effect. Yeah. Important concept here is this therapeutic index, and this is basically the ratio of this ED50 values. So it's the ratio of the half maximal value. So if you divide here like the adverse effect, they would have like a half maximum a potency of 10 microgram concentration. 10 microgram, it's not a concentration but a dose. And at 0.1, you would have like the beneficial effects, and then you would divide these 10 by the 0.1. You would see like, okay, this drug has a therapeutic index of 100. So this is somehow like the ratio of these two is 100 away from each other. And the larger this value is, the better it is. Often you call this the ED50 for the beneficial effect, the minimum effective dose. So you want at least to give this dose. On the other side, you call this half maximum value of the adverse effects, like somehow the maximum tolerated dose. It's like you don't want to go above this, and you don't want to be below the minimum effective dose. You want to be somewhere in between, and this curve should be as far as possible separated from each other. Important concept is often like okay, you can't see that. Important concept is the placebo effect, which we often hear, and this is basically a deviation from the baseline effect, which is just because you are in the clinical trial or because you take a tablet and so on, even if it doesn't contain anything. And often these effects are pretty large, and the actual response, which you measure, is a combination of the drug response, your baseline values, and what you see in the placebo response. Like, examples are, for instance, here, like in the ABV, this is like a readout from a lung basically a lung function evaluation parameter. So you breathe in a bag and so on, and you see like how much volume you can breathe in the bag, and the higher that is, the better it is. But you see that you already get like big changes in this volume if you just give placebo. And and so basically the effect of the drug it's like large, but in reality, like the actual effect is just the difference between the placebo and the drug effect here of the Montelukast. This is often important because just because people are doing something, you see already a large effect. And we see this also a lot like with the ACE inhibitors, especially in changes in heart rate and also changes in blood pressure and so on, because it's basically the people are just measured and they're in a clinical trials. So you see a lot of this baseline changes. And that's also why it's always important to have a control group where you basically don't give anything and put them through the same procedures. Important thing like about the pharmacodynamics is like basically this time delays. What you often have is that you see the concentration here in black of a drug. In this case, it's ibuprofen, and on the other hand, you see like a delayed response in delayed biological response. Like for instance, here it's a fall in rectal temperature. And it's basically like, okay, you often take ibuprofen to lower the fever, and but it takes some time, and it takes around like 2 hours. Like basically, you get already the maximum plasma concentration around 1 hour from the pharmacokinetic part, but it takes around 3 to 4 hours until you reach the maximum biological effect or the maximum reduction in temperature. And if you plot this, the concentration, basically the concentration versus the fall here, you often see like this, what's called like hysteresis curves, but but you see this round curves. And it's not like that it's on a straight line. It's going up at the same time and down at the same time, but you have like this time shift. There are different causes for this time delays. One thing is like that you have delayed distribution to the site of action. This happens especially in the low perfused tissues. You can imagine like if you have like things like bones, you want to like a medication which is working in the bones, and you have like very low perfusion of the bones, it can take very long until the drug is really distributed properly in the bones. And so you get a time delay between, okay, in the plasma, the concentrations are already really high, but at the site of action, it takes some time until the drug actually comes there. Other things are like really the underlying dynamics of the system. Often you can use like a signaling cascade, you have to synthesize proteins, these proteins have to work, you have to do like some restructuring, or changes in cytoskeleton or whatever, and or even like just bringing down the temperature. There's like a lot of physiological processes involved in this until this in the end results in a reduction in temperature. There's just like time delays. It's not like basically you do a switch and then the temperature goes down, because you just have to dissipate all the heat and so on, and this takes time. There are two concepts here. The one is like often it's called like pharmacokinetic rate limited response. And this is the case like if the biological response is basically instantaneous, and then the pharmacokinetic is dominating the time response. For instance, like if you look at the inhibition of the ACE, this is pretty instantaneous. So the ACE is in the plasma, and the disinocule immediately inhibits this enzyme, and this is in the range of seconds and so on. And so in the end, it depends on the pharmacokinetic of the disinocule, so how fast is it appearing, that determines how fast the enzyme is inhibited. The other things is like if you have a pharmacodynamic rate limited response, it means that the pharmacodynamic is much slower than pharmacokinetic, and then the actual rate limiting part is like, okay, really the change in biological response. Like here's, for instance, one example of such pharmacodynamic rate limiting things. It's omeprazole. most of you know that probably. It's an inhibitor of the proton pumps in the stomach, and it's taken often like for acid reflux, also many other diseases. And what happens after you take omeprazole, especially your gastric acid production falls very fast, but it takes very long until it returns to baseline. And you see that here, like on the left, pharmacokinetics. So if you take omeprazole, it takes around half an hour until you have maximum concentrations in the plasma. Then if you look at the change in acid output, this takes like for a very long time. You can see here, like if you take a tablet, like if you take one tablet of omeprazole, it takes around 3 days to go back to baseline. So you see a very rapid inhibition of the acid output, but it takes very, very long until this goes back to baseline. So you change your acid output for almost 3 days if you took one tablet of omeprazole, despite the omeprazole being basically out of the system after 2 or 3 hours. And the main reason is here that the omeprazole really binds covalently very strongly to the receptor, and it destroys the receptor, and it takes just some time for resynthesization. So you have to resynthesize the proton pumps, because the proton pumps basically permanently inhibited by omeprazole, and this turnover takes just a few days. But you have other things like it takes very long, like things like the blood thinners in the blood, basically the blood thinner medications and things like affecting blood clotting. It often takes weeks. So you have basically the biological effects are often like in the range of weeks or even months. Look at the turnover of cells, like if you take things like APO and so on, that they change the amount of hemoglobin you have in the erythrocytes. Okay, this just takes like in the range of weeks even. But I think this is often like important to just grasp this, that even if you took the tablet and it's basically away, and you think like, okay, I didn't do anything anymore, it's out of the system, like the biological effect can be like long lasting and have some time delay. How can you account for such delays in computational models? Often what is done is that you basically introduce like some effective compartments and some delay compartments. Similar like in this absorption model where we did this delay chain where you put like different absorption compartments one after each other, and you can create like this time delay. You can do the same on the pharmacodynamic effect. If you want to have a time delay between the drug concentration and the actual effect, you can have like basically this delay compartment, and this corresponds often to the concentration at the effective site. And by that, by this linking, you get basically a time delay between the pharmacodynamics, the effect here, and the pharmacokinetics, like the time-dependent concentration. Okay, yeah, a few slides about lisinopril. This is just okay, it's not too much, but I think the important thing there's a few important things you have to understand, or like it's important to know, but it's definitely not very structured what comes now. It's just like things on slides, and I think it highlights some important effects, some important facts. So the one thing is like there's really like a slew of ACE inhibitors, and this is often like for medications that you have like actually you found like one component for one drug which works, and it somehow binds to a receptor and shows a biological effect. And then often like it's like the key is like, okay, to modify this drug a bit, that you get better pharmacokinetics or better pharmacodynamics. So meaning you want like nicer, like long-lasting effects, that you don't have to take the drug too often, so like longer half-lives and so on. Or you want like better potency, that you have to give less of the drug, or you want higher efficacy, so more, the drug should do more. And so often you find basically like certain patterns, and then you just change that. And examples for that are, for instance, lisinopril, which is basically a modification of enalapril. But you can see also like here, some of the other molecules are looking very similar, like the silazapil and the quinapril look very similar. And so often you have, like, for certain classes of medication, you have really a slew of different things, and they often are different in the efficacy, in the potency, so this half maximal value, and often also like in the side effects. And differences can be by like small modifications that you can have different metabolic pathways involved, and you have like can have different tissue distribution and so on. And this is often what you see here. And in the project before, we did the ramipril, and now we're looking mainly at the lisinopril. Interesting thing is that you have like a lot of different ACE enzymes in the different tissues. So you have like basically different variants in the enzymes in the plasma, in the lung, and also in the vessels, and they are all structurally a bit different. And your certain ACE inhibitors can inhibit these things differently. And for instance, here in this figure, you see like for some of these substances, like here lisinopril, or also, sorry, I don't know the name, but benazepril, fosinopril. Often also these are all named very similar. So it's like if you have like a slew of these substances, like they're often like called very similar. So here, if you have a pril substance, you can be pretty sure it's an ACE inhibitor. Yeah, and you can see that you have like this Ka. It's basically the equilibrium association constant. Like for practical reasons, it's basically it's this potency constant. So the lower the value is, the larger the potency. This means basically the value where you have this half maximal effect. And you can see this is very different for these different substances. And it's also different for the different ACE subtypes. You can see here, like you have in the lung, right atrium, left atrium, and so on. So you have different effects on the different ACE variants in the different tissues, but you also have like a different effect depending on the drug. So they are structurally a bit different, and depending on that, they have different affinities to the enzymes and have different, basically different dose response curves. You see this, for instance, here. This is like such kind of dose response curves for the different ACE inhibitors. Lisinopril is not in here, but okay, it will have a very similar behavior. And this is okay. It's a binding displacement curve. In the end effect, okay, the real dose response is basically just 1 minus this here. I think the important thing is that you see like the sigmoidal curves, and the sigmoidal curves are different for the different drugs. And this really is shift in potency depending on the different ACE inhibitors. And this is often because they just fit differently. Like, so if you have very small, like this IC50s or potencies, you have often a very good and very strong binding. So this is often related, like, okay, your binding affinities are basically the potencies. And if you don't have such good binding, or like the structure not so fitting so well into the enzymes, you often have like much larger IC50 values. Okay. Yeah, and then what studies are doing, this is just an example for studies. But what they are doing, they're then looking for biological effects. Like the biological effects, which we see in the ACE inhibitors, are often like this zoo here of things. Like there's often HR, which is heart rate. The MAP is the mean arterial pressure. This is basically like let's say if you would take the systolic and diastolic pressure and average this in a good way, you will get the mean arterial pressure. So it's like let's say it's an estimator for the blood pressure. You have things like cardiac index, which is an estimate for the cardiac output. You have like systemic vascular resistance, which is an estimator for the resistance of the vascular system. And, yeah, so a lot of these are like basically the PCVP is a wedge pressure of I think the pulmonary capillary wedge pressure, something like this. It's basically a pressure in your in your vessel system. This is a resistance of the vessel system. So these are all parameters to somehow try to characterize, okay, how is the blood pressure? And then, okay, you don't have to understand this in detail. I think it's just important that you already saw that, because this is appearing a lot in the different clinical studies, because people want some simple readout. And okay, the systolic diastolic pressure is basically the straightforward readout, but often you will find other things like, okay, heart rate, mean arterial pressures, and so on. And so what you see, like if you give this different ACE inhibitors, you see, okay, a clear effect on these parameters. Often you see a reduction here in the heart rate, and you see reductions in the pressure. Cardiac index is increasing, and you see like that the vascular resistance is going down. And then you see like these effects are pretty consistent, but you have like variability between the different studies. Okay. I think the important thing to take away is here, okay, you have a lot of ACE inhibitors, and you have a lot of different readouts you could look at. So blood pressure is one of them, but there's a lot of proxies which are related to blood pressure and are important for modeling blood pressure. And so we are also interested in this additional readout and additional responses. Okay, how is this looking on in case of lisinopril? So here's one example like this. In this case here, this is sorry, this is called lysine analog, but it's also like the lisinopril is basically the lysine modified enalapril. So, yeah. That's why it's also structurally very similar, but it's has a better pharmacokinetics and also a bit different pharmacodynamics. Yeah, so what we see, okay, if you give the we have to look at this B. So if you give this drug inhibition possible enzyme, you see basically two curves. The one curve is the inhibition of the plasma activity. That's probably this curve here. So you see this is even at very low doses, you get already like a very high inhibition of the enzymes. And then you also like get a very prolonged effect. So you basically have here this pharmacokinetics. So this is going up like after basically you see like an increase in the plasma concentration of lisinopril after 1 to 2 hours. And you can see that the pharmacodynamic effect is basically instantaneous, and it's very strong. And the very strong is basically because you can, if you have a look at the dose response curve of the ACE inhibiting, so this basically showing you, okay, how much of the ACE protein is really inhibited. If you give lisinopril, you see that at very low concentration, basically at 1 nanogram per milliliter, you already have 50% inhibition, and you often like very, very fast in the saturation. And so you can see here, like if you have a half maximum value of 1 nanogram per milliliter, you can imagine like, okay, if you go up here to the 1 nanogram per milliliter, you already have 50% inhibition. And this is what you see here. Basically, like if you have like a few molecules of this lisinopril in your system, you already see like a strong inhibition of the enzyme. And this is also like prolonged here in the end. And this is like this nonlinear, basically what I explained before, like with the other example, this nonlinearity of this dose response, that this is really like it's shows strong inhibition of the ACE at very low concentrations, and then you at typical concentrations, like you can see here, above 10 you have 100% inhibition. So like basically you're above 10 here in this whole range between 2 and 24 hours, you see almost 100% inhibition. Oh, yeah, around 90% inhibition. You can look at, and this is like very direct readout. And so basically, this is the lisinopril is inhibiting the ACE, and you could say like, okay, our pharmacodynamic readout is like how much of the ACE is inhibited. And this is pretty straightforward and pretty good to measure and pretty clear story. But in the end, you're like much more interested in biological responses which are further away from the initial like inhibition of the enzyme. This inhibition results in a lot of in changes of different other protein, like basically this angiotensin 1, angiotensin 2, and the ratio of the two, it changes the renin, it changes the aldosterone, and in the end, this all changes somehow the blood pressure. This was what we had like at the kickoff meeting, like, okay, this is all much more complicated, like the ACE inhibition is like pretty straightforward, but how this is all relating to blood pressure, it's much more complicated. And this is this whole cascade of, okay, what is happening after the ACE is inhibited? And one thing which is happening is like, okay, the ACE basically converting the angiotensin 1 to the angiotensin 2. And so what you see is like this ratio of the angiotensin 1 to 2 is just going down. So you have, that makes sense. Here you have angiotensin 2 versus angiotensin 1. So yeah, it makes sense. So angiotensin 2 is what is after the ACE enzyme. And so you have much more angiotensin 1 than angiotensin 2 if this enzyme is inhibited. So if you just block the step, then angiotensin 1 cannot go to angiotensin 2. And so overall, nothing will be in angiotensin 2. And this is what you see here. But here you can already see, okay, this is still like a nice dose response curve, but it's already getting noisier, like if you have this first readout. Okay, let's see. Yeah, this basically this part. Okay. And we go, I think first we go through this okay, first we go a bit through this lisinopril document. And this is actually where where I put together the information. So I did we did together this initial literature research, and what I did for the now started to evaluate some of these publications and try to figure out, okay, what is important data which we need for the computational modeling. And so that I went through like the things which look promising, but I didn't evaluate everything. But you can see like, okay, there's a lot of overall we have like, I don't know, around 200 publications on first side which are somehow related to lisinopril, and we have to figure out, okay, what subset of data is now relevant for us. And so what I do in normally like opening these publications, evaluate if there's something useful in there, and then putting the information in this document. Perhaps I can show this with one more. Which is this? I have so much things here. Just looking if I see another study I didn't evaluate. These are reviews. Okay, this could be, for instance, one paper here, like this is like the influence of lisinopril on urinary electrolytes excretion after furosemide in healthy subjects. So there's already a lot of, there's a lot of interesting things in here. This is opened. Sorry, I just have to figure out, okay, what I have here on my laptop. Okay, in a second. Okay, this is better. I can. Normally, what I'm doing to evaluate this applications is okay. First, I'm just going over it and like getting a general feeling. Okay, this is somehow related to lisinopril. Urinary electrolytes is definitely something we are interested in a readout, because this could be an important pharmacodynamic readout. Healthy subjects we like. I don't like that it's after furosemide, so we have to somehow figure out, okay, what was actually done here. Normally So normally I'm having a quick skim over the abstract here. Here, for instance, lisinopril or its matching placebo was given orally using a double-blind crossover designs. And 4 hours after lisinopril, furosemide was injected intravenously and urine was collected. Okay, so it's a bit okay. So we like the first 4 hours, but we don't like this additional injection of the other substance. And so then we look here. This is basically like some furosemide. We're not interested in that. This is basically here the pharmacokinetics. And here we see like some readout, like, okay, PRA is plasma renin activity, ACE is our ACE inhibition. Okay, and this is overall urinary excretions. Okay, so this is definitely too far away for us. And this is more like, okay, concerned about the furosemide and if there's some interactions between the furosemide and the lisinopril. And the problem is that we don't have like a only lisinopril application. So this is like, unfortunately, not very helpful for us. And so what I did then is eventually I have to say, okay, I checked that. There is, so this is priority. We don't use the data from that here because it's not so important. We'll just annotate this was a new. Basically, I did something like that. I would just see if we have like another. This looks interesting. Yeah, like this is pharmacokinetics of lisinopril in hospitalized patients with congestive heart failure. This is definitely of interest. But the main application of our model will be, okay, we want to build a model for the healthy case, for the healthy lisinopril, and then want to apply it to the renal impairment and the cardiac impairment, and this is often congestive heart failure. And these are the two subgroups which also are the main target groups for lisinopril. So this looks overall like, okay, it looks good. Hospitalized patients are often not so good because they're pretty ill. But here we can see, okay, we have like some nice pharmacokinetic curves of lisinopril, and we have an oral and then intravenous dose. So this is pretty good data. So what I normally do is, okay, I'm just trying to collect the information. So in this case, I would copy to the clipboard. Okay, let's see how this works. And then I'm putting this in this document. In this case, I would add it like in this, we have this under heart failure here. We already have like some data in this document. And I would put it under the study ID. In this case, it's like this till 1989. And then we would add the information here alphabetically. So like somewhere here, we need after. Here, I would just add this additional information so that we 1989. We don't forget that we have like some information for this heart failure and that we have some pharmacokinetic information. And so this is pretty good data for the subgroup, and it's a very nice pharmacokinetic. In addition, we have to see if there's more information in here. Here we see summary results for urinary recoveries. This is also really good urinary recovery data, and we see like some AUCs for the different ranges. So this is pretty good pharmacokinetic information that we know, okay, how much of the dose was recovered. And this gives us also like some estimates, okay, that we can see, okay, after this is, for instance, like really interesting information here that we see, okay, after an oral dose, around 7 to 26% are recovered in the urine of the dose, and after an intravenous, 75 to almost 100% are recovered. And this gives us some hints what's happening with absorption, and it shows us that a large fraction of lisinopril is just lost after oral application, it's most likely directly going to the feces. Okay, and this is basically what you have to do. Like you can go a lot through the literature, and then try to just get first a feeling of, okay, what is the information which is relevant for the computational model. Let's see what else they have. Okay, here are more time courses. Mean serum concentration of lisinopril in normal subjects and in patients with congestive heart failure. This is also nice. The one problem is this is data from another study, so we have to use the original data here, but it shows like the comparison between the healthy subjects and the congestive heart failure. And we see there's not a too big difference. We just see, okay, that we have slightly higher plasma concentrations here in the intravenous case, and they're all orally, like there's basically no difference, or not much difference. And this is a bit of indicator that most likely perfusion or cardiac output will not have a too large effect on the pharmacokinetics of lisinopril. And this is like what part of the model building is, like a lot like looking at a lot of these figures and trying to figure out, okay, what are the small hints you can get gain, and so that we can later use this information for the modeling. And this is a large part of, okay, you have to just do literature review and like putting things together and seeing which subset of data is relevant here. Okay, let's see what else. And then there's some conclusions, and then that's basically it. So this was a pretty nice study. We didn't have any pharmacodynamic information. It was just pharmacokinetic, but it was oral and intravenous data. So this was pretty good. And just giving the text in this case. Okay, I also checked the study. This is nice data. This is in human. This is human. There was data about pharmacokinetics, these parameters like AUCs, and we had also like data about time courses. And this is high priority because it's really good data. And we would add this to this possible list of data. So this would definitely be a data set which we will use for the creation, and that's basically where this subset here of studies then comes into play. And there's like okay, this is basically like already the things which look pretty good on the first screening, and there's lots of data which could be relevant. In this case, okay, we have to work with priorities, but in this case, okay, there was some pharmacokinetic information in here. There was no pharmacodynamic information in here. Like this is all flags later on when we have created the data, and if we have used it for model building and so on. So did we include this in the documentation? Can we upload this to the database? Can we simulate the data? Did we use it for parameter fitting and so on? Are there certain issues with the data or the evaluation? And then we also have normally like we have to characterize what subset of data is like in there, so that we get a general overview what's going on. So in this case, we know there's no healthy data in here. The healthy data came basically from this Behrman. We have the study here also. These are healthy controls, but we had data for cardiac impairment in here. We had data for lisinopril. We'll take the flag here. We had urinary data. We had data for lisinopril. Bring this up here. We didn't have any data in the bile, and we didn't have any readouts for our pharmacodynamic measures. The pharmacodynamic measures which often appear, like I said, it's this mean arterial pressure, it's the blood pressure, heart rate, cardiac output, the cardiac index, the sweat pressures. We have like weight pressure products, systemic vascular resistant, and then we have like more like the biochemical readouts, which are more directly at the signaling. Like this is more like all the here pressure and resistance and blood flow and things like that. So it's more like, okay, the actual physical changes in blood pressure. And then we have more like the biochemical readouts, which is like ACE activity, renin activity, aldosterone, angiotensin 2, and so on and so on. And as far as I remember, there was also no data in here. I will just quickly check again that I didn't miss anything. But normally, okay, we see here we have the pharmacokinetics in this curve. We have like some urinary recoveries and AUCs. We have some comparisons with this healthy data, what these controls are from Behrman and from Ulm. But these are two studies we already have. And otherwise, there's no data in here in this study. Okay, and this is what then appears here in this in this document, and you can see like this Ulm and the Behrman, they also appear on the list here. And this would be like one of us would then work on the study and try to get this pharmacokinetic information and this time process out. Because with this we would then fit the computational model. Okay, yeah, that's basically how this works and how this table here appears. And now the thing is like, okay, everybody has like to select one of these papers. Like, okay, well, this is already a pretty good pre-selection. And some papers are pretty complicated, other papers are pretty easy. Unfortunately, this is quite heterogeneous, but we're okay. It depends on what data we need out of here. Okay, so we'll just go back here to this overview and just quickly go through this overview document that you get a feeling, okay, what is in the studies. So a lot of things are like often what is really helpful is like if you get such overview tables which are from reviews which give you a general feeling, okay, what is going on. And this here is a really okay. Can you hear me? I think Zoom is really don't let. Okay. Can you see him? Okay, perfect. Now it should work again. Okay, what you get here in this table is important parameters which give you a feeling of this drug, and you get this oral absorption. It's a really important parameter, and this tells you like how much of the drug is really absorbed. And you can see there's only like a small percentage, like 25%, and around 75% are going in the feces. And that's also what you see down here. If you get a look at the excretion of lisinopril, you can see, okay, a large part is really excreted in the feces. And this is what something that the computation model must predict correctly, that you have a small absorption only of the drug, and a large part is going in the feces, and around 30% are recovered in the urine. This is also what we saw like just now in this other paper, that you see this big difference between oral and intravenous application. And because if you give something orally and only a part is absorbed, and the rest is going in the feces. If you give something intravenously, everything is already in the bloodstream, and basically everything is going in the urine. At least here for this case. Other important things are like really that you see, okay, we don't have any hepatic metabolism. And this is also like some interesting thing that lisinopril basically it's a pretty simple substance from a pharmacokinetic point of view, because you don't have any metabolism and you don't have any complicated routes. It's just distributing in the system and it's going in the urine in the end. So the pharmacokinetic model should be pretty straightforward, and we don't have to take care too much about metabolism, which is good. And we can focus more on the pharmacodynamic part. The good part is also like, and that's why you only see like lisinopril measurements. You don't see measurements of any other metabolites, because there are no other metabolites than lisinopril. The onset of activity, you normally have 2 to 4 hours. You have a duration which is around 24 hours. You have a large renal clearance, and and this is what you see also in this curve, that you have like normally an action which is okay. This is pretty fast. In a few hours, it's going up, and then but yeah, it's long lasting, and it's normally around. It takes pretty long, and after 24 hours, you have still substantial amount of the drug in the blood. Yeah, you see this also in other studies here is like, for instance, Behrman. And what you see here, it's a nice study where they did a dose response, and you see a pretty linear dose response here. So there's a straight curve somehow, and you see like these curves are just scaled a bit. It's logarithmic axis, but okay, the higher the dose, the higher the plasma concentration, but it doesn't really change the clearance, which is also very nice. This looks like a pretty good thing. Also here, like you have pharmacokinetics then in this Behrman study where they did something for 10 days. And what you can see that there isn't too much accumulation over time, which is also good. Like if you see that, like look at the later peaks, they don't get very high, which is also pretty good, which means like most of the drug is really out of after 24 hours. This is also what we see here. So this looks all from a pharmacokinetic point of view, this looks all very nice. Here, some data about AUC dependency on the dose. And if this is on a straight line, this is pretty good. And then this is similar to the curves we showed before. So the area under the curve is really how much area you have under this lisinopril curve. And okay, and this scales basically linearly. This is also what we saw, like above the curve, the higher the dose, the higher the curve. But this is pretty linear, and then it's pretty linear in this typical dose range. And this is also really good for us because it's just simple. It won't generate much problems in modeling that. And so there's like from the pharmacokinetic part, there's a lot of good news, and this looks pretty simple. And here's, for instance, like things after multiple dosing, you see, okay, you have a bit of increase. Last time I showed this multiple dosing studies, and okay, this can be really a big accumulation. Here, it's only because you have a little bit left after 24 hours, you get a bit of accumulation. You see like this curve here is just going up a bit, and then these peaks are a bit higher, but it's not a big difference. And this is also very nice, and it's also consistent to the other study. Other things we see in general, okay, this curve look all very, I would say look very nice. Of course, if you look at individual curves, we see a large variability. This was not so clear here from this curve because we don't have any error bars on them. It's also like often a problem. You see none of these curves has error bars, so it's difficult to estimate, okay, how large is the difference between the subject. But if you look at tables like this, where you measure like the Tmax and Cmax and AUC for different subjects, you see there's a pretty large differences between the curves. And that means, okay, overall this is consistent if you do the mean over the groups, but you have like a large variability between the different subjects. But this is okay, and we can deal with it. And this is what you see here is one with error bars. You just see, okay, you have pretty large error bars here. The error bars on the ACE response are much smaller, but this is also partly due to this highly nonlinear dose response curve. Okay, and yeah, this was this, showed this before. Yeah, here you see also like, yeah, you can see differences between different subjects. In this case, okay, you see different subjects which are healthy, young, healthy, elderly. So you see a bit of effect on the age, but you see a very large effect on the cardiac failure here in this case. Okay, here you see like a larger difference. And that you get high, like it's in line with the other data we saw from this till 1989 right now. So if you have cardiac impairment, you get higher plasma concentration of lisinopril, and that's most likely because, okay, you have reduced perfusion, and then you have reduced cardiac output, and then you also have reduced filtering in the kidneys. And the kidney is the main organ, like removing lisinopril. So this somehow fits together. And we would expect similar things if we see renal impairment, that we get higher lisinopril concentrations. And then so like a lot of things is now like, okay, we have to have formed this mental picture, okay, what aspects are important, and how can we explain this changes in the curves, and also get a feeling for, okay, how large is the variability and how consistent is the data and so on. But overall, this looks pretty consistent. Okay, here's one study where they really reported individual data, and then even with like if you see this error bars, you often it's really difficult to get a feeling for how variable are these curves, because this looks still pretty good, and you can see there's different lines. You have to see the individual curves to get a feeling for how in reality this looks like. And this is basically like how the eosinophil looks like for different subjects. And you can see like very large differences here in the plasma concentrations. And some just go very high and others go very low. And we can see this here also. This is like probably a large contributor to this large variability is the different fraction absorbed. We saw like a large amount go in the feces and it's not absorbed, around 75%, but there's probably a large variability in this absorption. And so like people who have like high plasma concentrations, they probably absorb much more of the drug than down here. Because we don't have many parameters which could change like this curves. It's definitely like this fraction absorbed. There's a bit of perfusion, but this is most likely okay that we have a large variability in the absorption of eosinophil. And this could explain a lot of the variability which we see in the pharmacokinetics. And in the end, probably also like then in the pharmacodynamic effect. Okay, more curves here. Okay, you see some more curves with the arrow bars, but overall this looks, the important thing is that this looks pretty consistent, so that the time scales are similar. So after 24 hours, we have something left. It takes 48 to 72 hours until everything is removed. And we see similar things here. After 24 hours, something is left. And okay, that takes quite some long time. Similar here. And that we saw that those response. Also here we see large error bars, but this are, I think, only there's 18 subjects. You can see similar things here, like we have large error bars with the things. And it's probably you saw the individual curves above, because all the 18 individuals look very different, and then this is just the mean curve which is coming out. Okay. Renal excretion and so on and so on. Yeah, let's go a bit to the pharmacodynamics, and I think the important part is like I said before, you can have a ton of different readouts, and here's like one example, okay, where they just looked, okay, how are things changing if I give placebo, if I give 5 milligrams, or if I give 20 milligrams of lisinopril. And is that like the most interesting thing, like what there is a change in systolic and diastolic blood pressure, and the mean arterial pressure, the SMAP. Now you can see, okay, here in this case, not much is happening. I don't know. Normally, like you would expect this going down a lot, but okay, this is probably not mentioned. Blood pressure is often not a good readout for a blood pressure medication, but you want something which is direct, or like you basically like that more direct effects are often stronger, and like the real effect you want to have of the drug, they're often like not very prominent. And there's like a recurring theme, but here you can see it. Like you see, you can probably see changes in certain down here in the. This is more at the biochemical thing,
but this is pre-treatment, I think.
This pre-treatment? Ah, okay. So okay, good point. So then it's pretty good. Okay, then this shouldn't change. Perfect. Okay, responding values, pre-treatment. Perfect. Okay. Yeah, here are the effects exactly after the treatment. Okay, well, makes sense. And we see like pre-treatment, we see, okay, they're pretty similar. That's what we saw here. Okay, they don't change, there are no big changes, and that's what we want, that the groups are at time point 0 very similar. Okay. Basically, okay, for certain things, we see just that the readouts are very similar. For instance, here for the heart rate, we see this 3 different groups. So we have the placebo, lisinopril 5 milligrams, lisinopril 20 milligrams, and we would expect for certain readouts we want a big difference. Like for instance, here for the mean arterial pressure, it would be good if we see a big difference. And here we see, okay, like some stars. So this is pretty good. And the mean arterial pressure is, like I said, it's pretty close related to the systolic and diastolic blood pressure. So here we can definitely see, okay, after 8 hours or so, we see the 20 milligrams, it's doing something. But if you just compare the placebo and the 5 milligrams, you don't see too much differences. But you see like, okay, you see some dose response effect here. Okay, other things like cardiac output and so on. Okay, I don't know. That's probably no difference. And heart rate also like yeah, it's often like things like that. It's like really I don't know. It's just okay. It's probably because just the people are nervous or like I have no clue or like yeah, it's pretty consistent what you see here, like that this first drops over the first hours and then it goes up. I have no clue what this is, but it has to be something with the study design because this is pretty consistent between the different groups. And that's probably because they, I don't know, they are here in the morning and then in the afternoon you have different heart rate or some circadian rhythm or whatever. I have no clue, but this looks pretty consistent and you can see differences, but okay, overall, most likely for our model, we would just say heart rate is not changing and this is just a straight line. And it makes also sense if you look on the scales here, this is between 60 and 70. And so this is not too big of a change in the heart rate. Okay, here you see other readouts. And these are maybe kinetics of brachial artery diameter. So this is more like artery diameters, blood velocities, arterial flows. And you would expect, okay, if this really has some effect on okay, well, there should be some changes in the resistance and so on in the system. You can see, okay, we see here certain effects on the diameters and on the vascular resistance. And this is also what is pretty known about the ACE inhibitors, that it just changed the overall pressure and distribution of the blood and things like that. And this is like probably one of the large effects here also for the change in blood pressure, at least in the acute setting here of the first 24 hours. Okay, this is another thing which gets more complicated. Okay, if what happens in 24 hours doesn't mean that has to happen like if you treat someone for 3 months, but okay, that's at least the acute things are like pretty clear. The other readouts we have here, this is carotid, other arteries, other blood velocities. Okay, and this is just, it shows, okay, we have a time dependency of some of the things, but you can always see this is a lot of things are pretty noisy. Like often what is still consistent is like this resistances, like here carotid vascular resistance, and we often see, okay, that's also known like a bit what the ACE inhibitor should do. They change basically the vessel diameters and they change the resistance of our different organ beds. By that you change somewhat blood pressure. So at least there's okay, there's some good indication that this is the case. Okay, so this looks pretty good. Like I said, if you look at more direct readouts, things are much more nicer. Like for instance here, if we really look at the inhibition of the ACE as a first readout, this is pretty clear, and we see a nice dose response. Okay, if you don't do anything, the ACE is not inhibited, and if you give the lisinopril in different doses, okay, we see a clear dose response effect, and this is pretty clear. Okay, that the activity here, the absolute activity of this ACE is going down. Okay. And this is also, we see this in the dose response curves. Okay, this is a pretty nice response. So the inhibition of the ACE should be pretty easy to model and then include into a model. Other things like renin and so on, we see also very nice changes here. Like here also like the percentage change and the pretty big effects. That's also always important, like to have a good model, or which is useful somehow. The effects have to be big enough, and like it's often like, okay, if the error bars don't overlap, this is, I would say, pretty good data for a computational model. So on renin, we see also like pretty strong effects, especially compared like, okay, placebo, nothing is happening, and in enalapril, okay, we see similar directions here, which just a different inhibitor. For the angiotensin 1 and 2, which is also very close to the ACE, we see also like a nice dose response curve, so it's also pretty good. And then for other things like, okay, the actual blood pressure, things are often much more complicated. So this is not so, I don't know. This like it becomes just very noisy, and I'm always not so sure what I'm seeing here. Yeah, but overall, you see, like if you look at things like that here, okay, if you have studies where you have like 3 weeks and six weeks of lisinopril treatment, you see, and you often measure the blood pressure lying and standing, you can see, okay, you go down from the 160 to 100 to the 145 to 84. And here in the, after 6 weeks, okay, you're still pretty okay, but you go down from the baseline, and this looks pretty good. I said mean arterial pressure. Yeah, in the short term, we see pretty good effect. Cardiac output and so heart rate, I would say, okay, there's probably nothing there. That's just change, like no real effects. And heart rate and so on is probably also pretty constant. Other things, yeah, we have some changes in the, what is this here? Yeah, this doesn't belong here. Here, down here. Yeah, there's not much data about changes in urinary ions. There's also like this other story. Okay, the one is, okay, we're changing vascular resistance via the ACE inhibitors and cardiac output and all this stuff, and change the pressure. The other is like, okay, we secrete the ions differently, and by that we also change the basically the amount of fluid in the system. But there's not much data about that. I found like one data set where they reported a bit of urinary sodium excretion. You see some differences, but otherwise this is not so important. Yeah, like I said, the two subgroups really interested in the end, like the renal impairment and the cardiac output, the impaired cardiac output groups. And you can see some big differences here. Like for instance, this is like I think this is here what it means. Exactly. Here you see the differences in lisinopril between the single dose and the multiple dose, and you see like some okay, like you see like in the, okay, for the like the renal impairment, this is not going down really much about after 24 hours. So like if we saw the healthy data above, it was really after 24 hours, okay, there was some remaining things, but it was not close to the maximum. And that's what we see here, okay, because we can't remove much from the system in the renal impairment case, and renal clearance plays a main role. This values are probably still pretty high. And then we see also this accumulation. Like if you just look after 10 days, we have a large accumulation because in renal impairment, we just can't put everything out of the system. Okay. But there's also like some nice data about the, okay, this is pretty small. There's some nice data about some pharmacodynamic readouts, like in heart failure. What you can see, okay, there are also like changes in the mean arterial pressure, this pulmonary wedge pressure, and we see, okay, if you get first dose, second dose, we see this stepwise going down. This is always a good sign if we see like stepwise responses and dose dependency of things. This is always a good indicator. This is like a clear effect of the drug. And here also, like I said, like the closer we are at the ACE, the nicer the effects are. So like all this angiotensin 1, angiotensin 2, angiotensin 1, the ratios, and the ACE activity and stuff like that, we can probably renin and so on. Aldosterone, we can model pretty nicely. The big question is, like it was the same for the ramipril model. How do we like? We can most likely model these parts very nicely. So the renin angiotensin system, ACE inhibition, how is lisinopril doing all that? So there's pharmacokinetics to this pharmacodynamic part. But then the big question is, okay, how do we go from this biochemical readouts then to these changes in the pressure and in the MAP and systolic systolic blood pressure and so on and so on. So this is a bit of I would say this is the big challenge here in this project, that we can get pretty far here on the biochemical readouts, but then actually connecting it to blood pressure and so on, this will be a bit challenging. Okay. Let's see what else we have here. Yeah, and here we see also like similar accumulation in heart failure and so on, and this is like okay, what we see a bit if we have accumulation of lisinopril in the system, like due to renal impairment or heart failure, we get also like a large accumulation after the multiple dosing, and this is like I think one thing that our model can definitely predict. In the end, okay. I think what I wanted to do today is like just give an overview, okay, how this data is looking like. And of course, now we have to go like you could see there's a lot of good and interesting data in here, and we have to now get this for the computational model. We have to get this data sets, the pharmacokinetics and the pharmacodynamic parts, which are most relevant, into a machine readable format that we can feed the model with that. Okay, and this is basically like this set here of studies, and I think what will happen now, I would say like we have around, you can see here, around 30 studies, like a few of them are like not very nice, but there will be also a handful more which is coming to them. So in the end, it will be around 30 studies and around 15 people. So everybody will probably do two of these studies. And like I said, like they are pretty heterogeneous. Some things are really easy. Like for instance, if you look at things like where's the key? Like for instance, the skin studies, they are pretty easy because this is more like a spectrometry paper, and basically there's a lot of nonsense in there which we don't care about, and the only thing in the end which we care about is basically this one curve here. So it's like there's a lot in there, but it's pretty easy to do, and this is something you can do in an hour. Like if you know what to do, you can do it in 1 or 2 hours. So it's basically one curve which is in there. Like other publications, such as this Behrman, you can already see like from the paper, it's much more complicated because you have basically you have like the dose response curves here. You have like various curves of the lisinopril. You have this multiple dosing things where you have to have this individual time points. Here you have like some pharmacokinetic parameters in this table. Okay, here you have like some important clearance relationships, more pharmacokinetic parameters, okay, more curves. Everything is pretty heterogeneous. A bit more time courses. Okay, and you can see overall you have 10 plots, like not all plots, like the full-symmetric plots are not important. But you can already see, okay, compared to the other paper, this is like something, okay, there's much more work to do, but okay, then you also will understand much more. With the first paper, you probably won't learn so much. It's more like, okay, pharmacokinetics looks like that, and there was so much subjects. Here it's more like, okay, there's a lot of important, smaller details, which are later becoming also relevant for the model. And I think this is a bit the challenge. We have to like, okay, figure out like, okay, how to assign this paper, and also help each other a bit, like so the people who get like easy papers and finish pretty fast, but are good in the data creation, they can help others a bit and so on. Okay, so okay, what will happen on my side? I will clean up this table. You can already have a look at that, and I will also upload this publications to the GitHub, so that you can all have a look at this these publications. I also I really recommend to do that, that you have a bit of a look at this document. Just scroll through that, and then you should also get access to the basically this PDFs and the PDFs for the data creation, and then we assign until next week I assign somehow students to that, and then next week I'm basically showing how to do the data creation, and then already the first course is starting, and the first course will on the first 2 days we basically will do most of the data creation and then building the model. So far so good. Are there any questions or I know today was a bit of I think you now have to get started and just try to figure out, okay, what is going on. Even if you don't understand everything, just trying to look at the figures and trying to get a feeling, okay, how does such data look? And that you get a feeling, okay, how are the readouts and the things we want to model? And I would say like the pharmacokinetic part is pretty straightforward and pretty good, and then the data looks good. From the pharmacodynamics part, or biochemical readouts, I like them a lot. They look really nice, and they look also very consistent with the Ramipil data I saw before. Going through the pressures and so on, it will be more complicated. But we also like the nice thing is like then in some studies we see already the nice changes in pressure and so on. Okay. Yeah. Please, if you have questions, just ask. Otherwise, I will send some information around for the course, and I try to re-record the things I did last week. At least this worked better on the laptop now. I'm not sure what's going on with the other computer, but I try to fix that. Yeah, the record, it should work pretty. I hope this works now. At least it looked like for me. Okay, if you have any questions, please ask. Otherwise, write me by email.